So, good afternoon, everybody. Today's my topic is on molecular mechanism of microbial infection. So, before I start the topic, can I know, like, somebody knows Koch postulates? So, Robert Koch was a German physician who coined that the infection occurs because of a microbial agent or some external organism. So, Koch postulated four theories that he said that the person who is infected should have a external microorganism in him, and those organisms, that is the first one. So there should be an external agent to cause that infection. So the second postulate was that the organism should be able to be extracted from the infected patient, and the third one was that the organism extracted should be it should be cultured, it should be multiplied externally also, and if you insert that organism into another healthy person, it should cause infection again. And the fourth postulate was that the organism which we extract from the second infected person, that is the healthy person, so that organism and the primary organism which you extract from the primary patient, the both organisms should be the same, and it should be in similar, like all the characteristics. So that was a Koch postulate. So for infection to happen, there are three microbial factors. So the microorganism should inhibit the host immune system, one, and the second one is the it should alter the host tissues, so there should be any reaction to the host tissues. And the third one is that it should interact with the microbiota. So as Bhavit said from the last class, there are trillions of microorganisms in the human body. So the external microorganism should interact with the microflora of the host cell, and also it should interact with the products of it also. And next, the process of infection. So the process of infection is divided into four parts actually: colonization, establishment of the infection, disease progression, and transmission. So there are four stages. Starting with the colonization. Colonization occurs with the help of live pathogens, and also toxic pathogen products can also cause infections. Not only live pathogens. So live pathogens can be like mitigated with the help of airborne, that is droplets. So droplets, as we know, it is like it can spread through break in the skin also, and also through the respiratory pathway from your nasal to the bronchial tree. So it passes through the sneeze, cough, and talk also. When people talk, so that can also cause the spread of the disease. So the sneeze will mitigate around 40,000 particles. In per sneeze, 40,000 particles are released. So we can know that, so 40,000 out of 40,000 particles, there can be many microorganisms along with it. And the cough will release 3,000 particles in the air. So, and through the airborne, TB occurs, TB, influenza, measles, and as we know, COVID-19 spreads very vigorously. And GI and genitourinary system infections, those are mitigated with the help of contaminated food or water, and in unhygienic lifestyle also. The infections are UTI and cholera, which are spread throughout the and to like for the infection to occur, it's not like little small amounts of bacteria entering into our system can make a person unhealthy or infected. So you need a little minimum amount of inoculum. So the minimum amount of viral particles or bacteria colonies which will mitigate into an infection state. So large inoculum, it means that a large bacterial load should be there to mitigate into an infection. It should progress into an infection, so it needs a large inoculum. So that is a 10 power 8 particles are needed for Vibrio and Salmonella infection to occur. As we know, in India, we are mostly like TB carriers. So we don't have symptoms of TB because it is present in a very small quantity. So inoculum is not enough to progress into an infectious state. Whenever there is an immunocompromised state, then it progress into a very severe TB cases and all. And some infection needs only small inoculum, like Shigella, that is 10 power 1 or 2. Like 100 or 200 particle, 100 or 1000 particles also can cause Shigella infections in the colon. So that is colonization and establishment of an infection. So bacteria also has a tissue trophism. Tissue trophism in the sense that bacteria will not attack whatever tissue it is attached on. So it acts on a specific side itself, specific tissue itself. It will like proliferate in a like specific site of infection itself. Like as we see this group A streptococci, it will enter into the pharyngitis and soft tissue. It causes only pharyngitis and soft tissues, not the pneumonia, because it has a specificity. And again, Shigella species will cause infection in the intestine and not in stomach. Although it will pass through the stomach, but it won't cause any infection in the stomach. Rather, it will cause only infection only in intestine. Similar way, we know Helicobacter pylori. So it will cause infection just in the stomach region itself because it has a like a favorable environment over there. Then again, droplets. So droplets are the carrier of most of the infections, and it will it will pass the infection to the bronchial tree and trachea and alveoli. So hand touch to face. So whenever we have like some, we will be touching here and there. So our hands will be prone to many like microorganisms. Whenever we touch it to the face, it will like the microorganisms will reach into the like pharynx and inner regions of the nasal passage. So that will cause the infection. Again, environmental factors like bile salts in the gut. So presence of bile salts will favor the it's a favorable environment for Vibrio chlorella. So and H. pylori. So H. pylori, it releases an enzyme called urease. So this urease will convert the uric acid into ammonia. So we know ammonia is a basic in nature. So our stomach is an acidic medium because it will stop the infection actually. So this ammonia will neutralize the acidic environment of the stomach and thereby increasing the pH. It will cause tissue damage, change in the GI tissue, especially the epithelium, and there it will form colonies and form infection. Then bacterial toxins. So by heating methods and sterilization, most of the bacteria are like destroyed. But especially bacterial toxin doesn't get destroyed actually. So bacterial toxins are mostly heat stable, heat labile. So toxins. These staphylococcus toxins are produced in the dairy products and meat and salads, which are not like preserved very nicely. So they will release staphylococcus will release toxins on those products. Usually, what we do is we just heat it and we will just consume it many times. So the heating will destroy the toxin bacteria, but it won't destroy the toxins actually. So those toxins can cause staphylotoxins are enterotoxins. So these will cause like discomfort in the intestine and it will cause vomiting and nauseations and all. Then Bacillus curium. So it is the same emetic, like it is an emetic enterotoxin, and this toxin is present in the rice and starchy foods. Then botulinum toxin. So botulinum toxic are like produced by Bordetella. Yeah, Bordetella. So these are the neurotransmission blockage. Usually when they enter into the system, GI system, it will cause nauseation and vomiting. But when they enter into the systemic circulations, these will inhibit the neurotransmission and it causes like neurotransmission blockage. So later on, the symptoms are like delayed muscular contractions and the person is not able to function properly. So the bacteria, this is till here we know the bacteria comes and enters into the body. So now the attachment. So attachment is through three routes: pili, flagella, and autotransporter proteins. Pili is a hair-like structure which is present around around the body of the bacteria. So through pili, it causes adherence and anchoring to a specific cell. So they have a specificity. Each pili has their specificity. So and flagella helps in the movement of the bacteria. So through the like liquid membrane of our body, connective tissue and all, so they will pass through the this connective tissue with the help of a flagella. It's like a motor. So with the flagella they will travel. Then auto-transporter proteins, these are specific proteins which are present around the virus or the bacteria, which helps in the attachment to the specific site. So pili are again of three types, type 1, P and 4. So type 1 pili is mostly present on the E. coli, which has a specificity to the epithelium of the urinary tract, this thing genito tract actually. Then flagella helps in swim towards the side. It is mediated like towards the site also. It has a specificity how, in the sense that it is attracted to chemicals. So it is a chemical attracted like transport. Then human surface. So we know that Bordetella, whooping cough. So autotransporter proteins. Bordetella has a specific autotransporter protein on the surface of its like cell membrane. So this will cause the attachment to the like alveoli of the lungs. So that will cause a whooping cough. Then human surfaces. So human surfaces are like our human cell is mostly made up of glycoprotein or the other or else glycolipids or some carbohydrate membrane or mucin. Mucin is a mucous membrane. So what is the mucus is made up of mucin actually. So CFTR gene. This is a specific gene which causes cystic fibrosis in humans. So presence of this gene causes cystic fibrosis. Thereby, the cystic fibrosis increases the susceptibility of the infection. Again, this FUT2 gene, this gene helps in the mucin D arrangements actually. So the mucous membranes, how much the mucus should be there, how much, what is the layer and all. So that is regulated by FUT2 gene. Any presence of two unfunctional alleles can cause the dearrangement of mucin, thereby decreasing the mucus layer and thereby increasing the this thing glycan layers of the cell. So mucin is actually a protective agent. So it will stop the entry of the microorganism, but leaving the mucus when dearranged, so it will expose the glycan layer and thereby increases the susceptibility. Again, this GM1 is a surface protein which is prone to Clostridium and H. pylori. Then OMCB is the for E. coli again. These are the surface protein which helps in the attachment of the microorganism to the cell layer, cell surface. Then replication. Of course, the microorganisms should replicate. So replication is like some organisms will be replicating outside the cell membrane itself, the human cell, host cell, just outside the layer itself. Some will go internally and they will replicate inside. Like we know HIV will replicate inside the human cells. And for the surface, Pseudomonas species will usually replicate on the outer layer of the human host cell. And intracellularly, again, there is a Salmonella. There's one more example. There are many examples, but I just taken one. And biofilms. Biofilms in the sense is that when a cell, the microorganism infect a cell structure or a tissue, so it will be on the surface itself. So on the surface itself, it will form colonies. So on colonies again, there is a like high chances that macrophages will come, our defense mechanism will come, and like it will stop the colonization. So these cells actually, Staphylococcus aureus and aerogenes, so these will form a biofilm over the colonies, so that the macrophages and other PMNs, the polymononuclear neutrophils, they will not recognize these colonies and they won't find any inflammation and nothing. So these will grow by forming biofilms on the tissue sites. So then again, microbial entry into the cell. So how will the cell go intracellularly? So there can be two methods, trigger method or zipper method. So trigger is usually the example is E. coli, and for zipper it is pseudo TB, tuberculosis. So trigger method in the sense is E. coli will have an injection like a organ on its outer surface. So what this surface, so what this will do is it will release a specific biomolecules which will in turn get into the cell, and this will trigger the cell membrane to enclose the E. coli inside. So these chemicals are mainly mediated to enclose this E. coli into the cell. so that the cell can the E. coli can get into the cell. So this will be the vacuole actually. Okay. Then zipper method. Zipper method means it will have various like pili on the outer surface. So this will actually push through the cell and get into the host cell. It will be more like a pushing, and this will be like more like a chemical mediated. Then again disease. So disease mitigation is uh, inflammation and phagocytosis should occur so that the disease can be like looked or observed on. And so inflammation will be through many cytokines and neurokines which will be released by the inflammatory media through the inflammatory mediators. And the molecules in the human body like collectins, surface protein A and D, these molecules will tell the phagocyte cells that this is an foreign substance which should be destroyed and which is not acceptable in the body. So these are the proteins which will help in the recognition of the like foreign molecules by with the help of phagocytosis and PMNS. This will destroy the foreign organisms. And negative bacteria, negative gram negative bacteria will have lipopolysaccharide bilayer on the external side, and positive bacteria will have peptidoglycan layers and viral particles. These will attach to the CD4, and these all will cause inflammation thereby. And some organisms will have antiphagocytic membrane, means that it will stop the phagocytes to recognize the foreign molecules. It will just like fool the this defense mechanism that no, it is our own host cell, it's a human cell only, there's no problem in it. And these type of organisms are cryptococcus and bacillus organs. And endotoxins, these organisms will produce endotoxin, thereby which it will cause fever. Fever is a defense mechanism of human body itself, but the endotoxins will trigger the fever actually. So fever, muscle proteolysis, uncontrolled intravascular coagulation, and shock. So cytokines will be the mediators in between the endotoxins and the symptoms to be occurred. Then again transmission, as we know, when the cells will divide again, they will travel to the if it is a pneumonia or some TB or COVID-19, when you cough, then it will spread again. That is the process of infection. That's all. Thank you.